back till September 8th. Yeah, Cobb, I've seen a lot on the news, August 17th, but still doing the in-person or virtual by choice. Oh, Lisa said teachers go back on July 27th.
But we'll see.
Let's see. Marietta's going back on time. Yikes. Yeah, still options for Athens students. Amanda said going back on the 4th with only 200 signing up for virtual. So everybody wants to go back. Yeah, Kristen said Gwinnett delayed until August 12th. So I don't know. Just don't know. Oh, good, it's recording now. Thanks, Kristen. All right, so we'll go ahead and get started, and let me find my little PowerPoint share with you guys. And I'll do like I did last time and just leave it kind of in the typing mode so I can type on it. Because basically I just wanted to get y'all to brainstorm today. So I started with a couple ideas, and then hopefully that'll get you guys thinking, and we can help each other to come up with some ways to incorporate. So we can do a quick intro and just say who you are, where you are, if you want to say what subjects you taught. I think I caught everything that everybody teaches down here below. So we'll go ahead and go through. Kristen, do you want to say hey?
Had to unmute. Hi, good afternoon, not morning anymore. Welcome, I'm Kristen. I am the Director of Programming at the Georgia Bioed Institute, and I'm glad to be here today. And we're super lucky to have Megan leading everything through Innovate Bio. So thanks, Megan.
Well, thanks. Just trying to help out.
You're awesome.
Oh, thanks. Amanda?
Hi, my name is Amanda. I teach at Marietta High School. This fall, I am supposed to be teaching biology, applications and essentials of biotechnology, and honors chemistry.
Yeah, 4 preps.
Yes.
Yeah, that's the life. Oh, well,
I'll be
hopefully we can
thrilled.
help you make things easier.
Well, we're teachers. We make it work.
I know. Well, I'm here to support. We are here to support you. So.
I appreciate it.
All right, Jennifer.
I'm Jen. I'm out in Cobb County. I teach a variety of sciences and computer science in the biotech pathway.
Awesome. Thanks. Julie.
Hi, I'm Julie. I teach in Athens at Cedar Shoals High School. Thank you, Megan and Kristen. We appreciate what you're doing. I teach agriculture, and under that umbrella, I teach plant science, animal science, vet science, nursery landscape, and basic agriculture. And I also incorporate the bioscience into FFA agri-science fair competitions, which we've had a lot of success for.
Awesome. That's interesting. We probably want to know more about that and pick your brain moving forward, because we've gotten the ad to do more agriculture-based things. That was the request from the DOE. So love to pick your brain sometime. And thank you for participating in all of these. Lori, next.
Hey, I'm Lori Howard. I teach in Cobb also at Walton. And for the fall, I'm teaching college prep bio, honors bio, and maybe an applications to biotech. Not sure yet.
Yeah, it depends what the numbers look like, I guess.
Yeah.
Yeah. Well, hopefully.
Yes, hopefully.
Thanks, Lori. Lisa?
Hi, I'm Lisa. I teach at Griffin High in Spalding County, and I teach 12th grade environmental science.
Awesome, thanks. And I think Stan is last.
Yeah, I'm Stan Harrison. I teach the biotechnology pathway at Morgan County High School at their Academy of Science and Technology.
Awesome. So thank you guys all for coming today. Okay, so we already talked a little bit about what's happening in your districts in the fall. So hopefully, we can support you moving forward. We're going to talk today a little bit, I want to brainstorm with you guys about how we could incorporate different biotech labs into other courses. Obviously, one of the keys to learning is repetition. So if we could come up with some ways, and I have a couple ideas, but some other ways that we can kind of pound the same ideas and the same techniques into the students' brains, that would, that's going to help them in the long run with learning these skills, learning the essential skills that we've previously mentioned are important for employers. So I kind of broke it down just by the subjects because that's how my brain was working, but we can add to this PowerPoint and just kind of throw some stuff together as we go. I did start a resource folder and I'll share that in the chat. So if you have any labs that you want to share, feel free to dump those in there and I will try to remember to, well, I'll save the chat, but I'll go back and check our folder and add things as I think of them. You're gonna not let me use the chat today? That's nice. Okay. Maybe I'll send it to Kristen and you can put it in the chat because my computer is like, we're not doing the chat today. You're presenting or you're chatting. You're not doing both. Okay. All right. So I started with chemistry. So we've done the Kool-Aid chromatography a good bit, and I'm super familiar with that one, and chromatography in general, but you can definitely use that in chemistry to talk about different separation techniques. Especially the Kool-Aid chromatography relies on polarity and using different concentrations of alcohol, isopropyl alcohol, to push the different dyes through. So basically they have more affinity to the column, and then depending on what concentration of alcohol you push through, you can push the molecules of dye off of the column and separate out, say, purple Kool-Aid into the blue and the red. You can use gel electrophoresis to separate dyes. Again, those would both fit in super well in the separation techniques unit. So talking about separating with an electrical field, determining the charge of particles. So you could have them put the well in the middle of the gel and then see which way things travel in the electrical field. Do you guys have any other suggestions? And I really want to know where my chat went.
Are you wanting physical or virtual labs?
Whatever you guys have, that's a good point. So at this point, I think we need virtual resources.
So I've been putting together a list of virtual resources, and it has anything from learning how to use a pH probe, which is both biotech and chemistry, to molarity and lab equipment, lab safety. There's different tests like oxidase and catalase tests that could go in for chemistry.
And is that like a folder that you can add to our folder, or?
It's a Word doc, so I could add it into it. I'm trying to see if I could share it so y'all could view it, but
I can stop sharing if you want to view it real quick.
Okay.
Okay.
Do that.
Stop share. So I think now you can share.
Okay.
Should be able to.
Is anyone able to share, or have that blocked or something?
I think so. I could click the share screen before I became the host.
Okay. I meant actually the chat, so that Jen could drop it in there maybe.
Oh, Amanda.
Huh?
Oh, here we go. We're screen sharing.
Okay.
She was saying you could drop it in the chat.
Yeah, I could do
oh,
that.
Cool.
Yeah, so it has a little bit of everything. I've been trying to break it down. So this is just all biotech stuff. But you have anything from job skills to lab math, graphing, metric conversions, which you use in both chemistry and biotechnology. Microscopy, you don't use too much in chemistry, but you do in biology. PCR, we don't do teeth, but gel electrophoresis. Yeah, you could talk about the chemistry in DNA extraction. And yeah, so it's just all kinds of, I've just been putting it
Awesome.
together so I can go through and look how to use analytical balance. So yeah.
Nice. Yeah, if you could share that into the folder, that would be awesome.
Yeah, I can do that.
Cool. And then I put a link in the chat. So I had been compiling some teacher resources, and some of them are online labs. I see a lot of yours is NC Bio Network. The Lab Exchange has a ton of good ones too.
Yeah.
And they're free. You don't have to create a login for theirs, but if you do, I think as a teacher, you can make specific playlists for your students.
You can, yeah, do a complete pathway. I've got some of the main sites down here.
Oh, yeah. Cool.
So, okay, let me see if I can now unshare. Let's see, going back, maybe stop sharing. There we go. Okay. Sorry.
Yeah, perfect. Thank you.
You're welcome.
Awesome. Okay, so I'm going to reshare just so we can all be on the same screen. Okay, so Amanda shared links in the drive. Cool. Yeah, so online labs, definitely, those are super helpful. You guys also may have the option to do some demo stuff, depending on whether your school lets you back into the school or if you have to do it at home. You know, maybe you could take some equipment home. I know Janet Standevan has taken home her PCR stuff because she has a mini PCR machine. And so she's been running PCR for her students and then just giving them the data. And so then they can still analyze the data and follow along that way. So that's good. Cool, thanks, Amanda. So biology, I threw stuff down. I think I put chromatography on most just because I copied and pasted. But you could go through chromatography and the different uses as far as separating out DNA, separating out proteins. Gel electrophoresis, again, so separating samples from DNA, how it's used in conjunction with PCR to confirm samples. PCR, you can talk about the different uses. So like any kind of detection, like we're seeing now with viral detection, you can use it for bacterial detection. Basically any kind of detection that you want to do of looking for a specific DNA sequence. The lab that I was working in at Georgia State did a GMO unit, and so they looked for the 35S ribosomal subunit DNA to confirm the presence of GMOs. So anything else on that? And I keep losing the chat. But I also put down protein isolation. So you could look at the different ways to isolate that. I know we talk about the DNA extraction all the time and how easy that one is. But those are some other options. Bacterial transformation, so you can show that as far as how DNA, you can introduce new DNA into organisms and really go over the central dogma that way. Especially like the Piglo kit, if you don't want to get into kind of all the arabinose stuff, you could focus on just when you turn it on, then it makes the protein and show the DNA to RNA to protein that way. You could look at microbial growth, exploring microbes, plating microbes. We had talked about maybe there's a way to do that as an online thing where you send everybody home with a plate and just give them specific instructions about, okay, when you're done, you have to show me that you have bleached it or, you know, run alcohol over it or something. So just giving your kids something to work on at home if they are stuck at home. Can you guys think of anything else that you would incorporate into a general biology class that, I guess mine were 9th graders when they took it. Clayton, it was 10th grade, but that 9th or 10th graders would be interested in? Everybody's muted. Again, losing my chat. So if you guys think of anything too that you want to share, feel free to go back to the drive and share that at any point. Okay, AP Bio, I legit did not put anything. See, I stopped right here. You can go over the same stuff as biology. The Piccolo lab is one of the big labs anyway. We were talking about between biology and AP biology, really, because things are moving more into biotech, there are a lot of areas where you can use those labs to investigate the topics. So the same ones I listed in bio, just going an extra level with them. Environmental science, I posted Trisha Peterson's eDNA lab on that drive, and then one of them is, it says like recipe, I think. That's basically the teacher guide, so it has what dyes you would need to do in each tube, and then I think it has the recipe and then what the result should look like underneath. And I got the dyes for that, which I can add it back. Stan had added it to a different version that he shared, but I found a link on Flynn Scientific. They have a process to dye for DYE4 kit, and they basically, I took the dyes from that and then used them for this lab that Trisha came up with. And so what she does is chromatography. So what she does with the eDNA is uses the dye samples, and then you aliquot them out, and they run to represent the different samples, so at the different testing sites. So a certain banding pattern would represent the presence of an invasive species in that area. So you could have them do the dye lab first, and then if you're able to do PCR, you could go look and try to confirm the presence of some of those samples. So chromatography here too for environmental science.
I'm also going to do through the Rural Science Initiative, the Piccolo lab. We've been talking about GMOs in my environmental science class.
Oh, cool. Yeah, that's a good point.
Because people don't really, I mean, the students don't really understand the concept of a GMO. So I think that's an excellent visual.
Yeah, so you can show them how to actually genetically modify organisms.
Mm-hmm.
Cool. Yeah, that's a good one. So Jen, are you unmuting?
Yeah, we do biodiversity surveys for environmental through, we have them collect plants, or some of them do insects, and then we do basically DNA barcoding. So we'll send them out for sequencing and stuff.
Cool. So they can try to identify it first, maybe using a book, and then they can get some confirmation by sending in the sample for DNA barcoding.
Yeah, so they have to pick one plant, like we do quadrat surveys, and they pick one plant that they're unable to identify, which unless they're flowering, they can easily find at least one that they can't, and then they do some isolation, and then I have my upper level kids do like protein or DNA purification, and then we send them out for sequencing.
Okay, cool. So, so you have multiple classes kind of working together on that.
Yeah, because my environmental science is for 9th graders, so they can do the DNA isolation real easily, but they're not always as precise to get really good enough samples for purification. I have my other kids do the PCR for it, and then my 9th graders go back and blast it and figure out what it is.
Awesome. Okay, So yeah, having other classes help too. That's one thing that we've talked about. I know Tima Hoskins out in Paulding County had her kids do the eDNA lab, and then her AP Bio class liked it so much that they went back and they rewrote the lab for her chemistry class and basically made it into a whodunit thing instead of the eDNA. So making their own forensics lab out of it. So the kids get really into helping each other. So that's a great way, right, to have one class help with the other class's samples. Cool. Anything else that you guys can think of? Some good ideas. Thanks, Jen. So physics, I put down chromatography. So again, you can, I think I just copy pasted, again, chromatography everywhere. But getting them to look at, you can look at the charged particles from that perspective. Gel electrophoresis, you could have them design their own STEM boxes. So Biorad sells the kit, but I've been getting stuff off of Amazon. I bought these little, it wasn't soap dishes, it was like jewelry dishes, and they're a nice size. I made combs out of that craft foam. So you get the whole pack of craft foam at Michael's that's like 8.5 by 11 inch sheets, and they're kind of thin. And so I cut out the combs to fit those. So the students could design their own combs. They can, you basically just show them how to pour the gel, and then they pour it, and they can maybe try different voltages to see how things run and look at the relationship between voltage, current, and the movement of things in their STEM box. So next thing, I know Julie teaches all the ag classes and animal science. So just some ideas for that with PCR. You can use that to identify anything, like we said, GMOs. Somebody was telling me the other day, chicken industry, some of the ones that are more biotech focused use basically confirm the presence of bacteria using PCR. So instead of trying to like plate a sample and get it to grow that way, PCR is a lot quicker. And again, like we were saying with environmental science, to talk about GMOs, using that bacterial transformation to describe and kind of give an intro to how genes are inserted. Yeah, viral detection too. So identify viruses and bacteria. Okay. Any other thoughts on that? So basically
Yes.
any of the genetic modification. Julie?
Yeah, I have a suggestion.
Yeah.
There are these seeds that you can get. It's called Wisconsin Fast Plants.
Oh yeah, the fast plants.
And they get up to reproduced size within 2 months, or you can
Nice.
cross-pollinate them. So you can breed the plants and then run the DNA and compare those. Also, using different influences on the same type of plant. So if there is a beneficial bacteria, we did a lot of research on what's called smoke water. And you water the seed. It is water that has been saturated with smoke from burning plant material. And that actually encourages seeds to germinate faster. And in some seeds, it actually encourages germination. They will not germinate until they have the smoke water present. But there's a lot of research. There's some research being done at UGA on it. And one of the things I want to try to do is to run the DNA to see if the smoke water actually has an impact on the DNA of the plant, long term.
Awesome. Yeah, and maybe it's turning on specific genes.
Yes.
Yeah.
It actually is in the roots from the research that they're doing at UGA, that there's signals being sent out.
Awesome.
To the roots from other plants. And studying insects.
Yeah.
We partnered with USDA this past year, and we were sent insects What was it? Yeah, I got a shipment in of like 60 of them in a certain phase of their life cycle, and we were able to watch them grow and develop and then run little labs on them. We
Nice.
did not get to the biotech part, but it would have been good to phase that in farther on in the year. But
Yeah.
it's
I
cool
guess
for them
that
to
got
watch
shut off
life
'cause of-
cycles.
Yeah, that got shut off because of COVID.
Yes.
Yeah, timing. Cool. So is that something that you're able to request through them?
Yes, it is. You it's the corn earworm. It's the tomato earworm and the corn earworm, and you have to get an e-permit through USDA, and then you email them and tell them how many of them you want, and they will put them in individual containers with their food source. And they're little bitty plastic cups, like 2-ounce cups, and they put them in a box and ship them to you. But you also have to go through the little education about not releasing them out into the wild. And
Okay,
you have
yeah,
to
that makes
freeze.
sense.
You have to freeze them when you're done to kill them.
Right. Proper disposal.
Yes.
Awesome. That's so cool. Okay. Anything else?
Yeah, you can also do, it's not like strictly biotech, but it's really good because the kids get experience working with different species and stuff. And it's, we don't get to work with animals that often, but you can do, along the same lines, you can do LD50 labs. You can do them with brine shrimp, but it's not very fun. I like to do them with ladybugs, and the kids get practice doing serial dilutions with, you can have them choose different like over-the-counter pesticides, and you can determine whether what they say on the pesticide is actually accurate. So most of them will tell you like what percentage of pests it should kill. I worked in an entomology lab in college, so I like the kids to work with, do things that I actually did, and they learn how to apply the pesticides to the ladybugs and run LD50 labs and things like that. So it's not super biotech, but it's nice because it incorporates those biotech skills to other things.
Yeah, and that's definitely a good way to introduce LD50, because that's a concept that they're going to see if they're looking into like toxicology and things like that. So
yeah,
that's
really
awesome.
fun.
You can also not only with the insecticides, but you can use other types of pesticides like herbicides.
Yeah.
We did a competitive research project on the impact of specific herbicides. Roundup is in the news a lot about carcinogens, but we put herbicides in soil to see what the impact was on earthworms.
Yeah. Okay, that's a good one.
That's an environmental one. That takes a long time to study, but you can get earthworms off of Amazon, and we use little plastic shoe boxes as their habitats, and then get bagged compost from a store to make sure it is all, we got it from Lowe's to make sure that it's all uniform for their bedding.
Yeah, that's a good point. Cool. So soil.
And another thing, I don't know how difficult it would be, we haven't gotten this far yet, but to study the actual bacteria in the soil in relation to the herbicide use too.
Yeah, and so my master's was in microbiology, and I did the environmental microbiology class, and our professor was big on studying soil bacteria. And we spent a lot of time talking about how you can make batteries from like the lake mud, basically.
Oh, yeah.
So, yeah, so they could look at the composition of soil generally in the bacteria, and then even you could extend that to talking about the herbicides or pesticides in the soil, and maybe the different chemicals have effects on them.
Yes.
So, and then see how the plants grow, depending on what you've treated the soil with.
Well, and also you can look at the underground environment, not just how well the plants grow on top, but how the roots develop underneath. You know, not many people are looking at the roots now. They're all looking about above ground, when basically you need to know what's going on below ground to support that above ground growth. So yeah, root development is a really big thing.
Yeah, so factors influencing. Right, and they could set that up as a nice controlled experiment and really use some of these biotech skills to set it up properly.
I actually saw someplace that somebody was using the old CD cases to start seeds.
To plant them sideways so you could see the plant and the root growth.
Right, and that would be real cheap. I actually bought some seed starting pouches where you see through them, and they were pretty costly. But those old CD cases that way you could actually see the roots developing, and you can potentially measure it.
Yeah, that's a good point. So maybe they pile the soil high enough that the roots have enough room to kind of grow down.
Yeah.
Yeah, that's a good point. It's kind of like the ant farm where you can see them. So
yes,
that's cool. Awesome. I like that. Cool. We have 2 pages of agriculture now. And then you can also take all of this back into your biotech class, y'all that teach biotech, and incorporate some of these other concepts that way. Awesome. And then I did animal science separately. So in addition to some of the stuff that we talked about with PCR detection of bacteria, viruses, etc., you could basically do genotyping. So like Jen was saying about the insects. So you can have them try to isolate the DNA, do some PCR, identify what the organism is. You can talk about it in terms of animal husbandry and like the species survival plans. So how they basically try to match up the best genetic diversity when they breed 2 animals together. So that's how they determine which pandas should be mating and things like that to introduce genetic diversity. And I don't know if the San Diego Zoo Safari Park has it online, but I did a professional development there where they kind of walked us through all the different work that they do in the conservation center, like with breeding the cheetahs and everything like that. So any other ideas? Julie, and then Amanda.
The animal science class under the agriculture umbrella, although it says animal science and biotechnology, really doesn't include any biotech in the standards. And I was trying to think of what would be, something biotech for those standards. And it would probably have to be on a small scale like aquaponics or poultry.
Aquaponics.
Yes.
I'm going to put animal science, quote, biotech course.
Yeah.
Yeah, so right, aquaculture, and Stan can probably speak to that. He does tilapia farming in his class. I don't know if he jumped off.
No, I'm here.
Hey, Stan.
Hey. Yeah, that's that's cool. The we're talking about doing virtual though, right?
Not necessarily.
I'm not.
Well, aquaculture is really cool. Everybody digs it. All the students really like it. It's it's our dog and pony show. It's our main attractor for my program to get students in there. But it's a lot of work. It's a whole lot of work. You have to have the right equipment. We actually got ours donated from the University of Georgia. You'll be amazed at what you can find just laying around in a lot of these colleges that are either older equipment or not used or whatever. So, I couldn't really pull it off without having a 550 gallon tank. And it's it's an enclosed system, right? It's a biofiltered system, so you have to have a biofilter, and you have to have an airlift. You have to have a pump, an air pump, to lift the water back from the biofilter into the tank so it circulates. So yeah, you can. I mean, you could actually make one of these if if you're really crafty and have a good engineering program at your school or something. Get them to actually fabricate these types of things. You can find online. You can get specs, although it's few and far between. I'll be honest with you. Specs on these things. I found it was hard to actually get replacement parts for my unit. Yeah. Yeah, go ahead.
Do you have yours outside in a separate building, or do you have it in your classroom?
No, I actually have a separate room. I call it my dirty room. I've got my clean room for all my genetics work with a hood, and then I've got a separate room with all my agriculture stuff.
Oh, okay.
So, yeah, I just put it in there. And you need to, just because there is a smell associated with it.
Yes. Well, mine's in a greenhouse. I built a greenhouse for mine.
Oh, yeah. Well, that's great. Yeah, perfect. And you
it's
just
small.
have to have, is it small?
Yeah.
Yeah, you could. I mean, I've seen people do it with koi pond type thing, inserts on a smaller basis. I raise tilapia, and they get big and they grow really fast, and they also foul the water. So I have to have a large tank.
Well, I mean,
cats are typically the same way. So
my greenhouse, I think, is a 16 by 8, and I've got a big system in it, so it doesn't allow for a lot of people to move around in the greenhouse. But
right.
This can be done on a smaller scale. I mean, if they wanted just to study the tilapia, it could be done in the classroom with just tanks, small fish tanks.
Well, sure. I mean, yeah, if you just wanted to have a couple, we're raising them. I mean, we're doing it as a project. I mean, this is just how I do it. It's like starting a little company. So I force my kids to do the research, to cost out everything, figure out how much feed we're going to need, put it on a schedule. So we have a project manager, we have a purchasing agent. And then at the end, we harvest and we have a fish fry, and they have to earn the money. They have to sell tickets and try to recoup the expenses to see if we can have a profit. And they quickly learn that it's almost impossible to turn a profit on aquaculture in Georgia. But hey, that's part of the learning experience too, is say, okay, so how much would you actually have to do? 100's not going to cut it. 100's a lot of fish for a small tank, but
are you growing any plants in association?
Yeah, I've done it before. I've had the insert on the top, which I don't like because it gets really nasty. And then the fish, of course, will eat the roots if any of them come through the rock wall or any of the type of inserts. So I think an offset hydroponic is a lot better than trying to do it in the tank itself. I think you could probably, if I really wanted to work at it, I could probably figure out a larger cage where the roots would have more room to grow into the water. It's great, great fertilizer. The plants grow like mad from the water, straight out of the water, out of the fish water. Yeah. But yeah, a simple offset sort of thing with a tray or a sink type of thing. And I think you could grow plants a lot more effectively. I've threatened to do that for a couple of years, but I've just been too lazy to get around to it.
We have 2 grow beds.
Okay. Yeah.
And they're the big feed troughs.
Yeah. Mm-hmm. Yeah, that's wonderful. And my problem is I've got to figure out how to recirculate that water back into my clarifier, is what we call it. The sink that actually allows the water that's siphoned up from the bottom. It allows it to slow down and all particulates in the water to drop out. So somehow I've got to have another air pump to pump out of the hydroponic plant sink into that. So, maybe it's a collaborative type of thing where I work with another CTA program in our school, maybe once again, our ag engineering class. If I can get them on board to kind of figure out some of the construction issues, it will allow us to fixate more on the science side of things. So yeah, I mean, I think it's a great idea. The kids really love it. You're showing a closed system. So that's another aspect I think is really good with this type of thing. You're showing an example. Okay, is it sustainable forever? Well, no, because we have to actually take out water every day and we have to put in new water every day. So it's not really a closed system. And then we can do water quality tests throughout. And it's really crucial at the beginnings of this when they're fingerlings. And we have to add some additional chemicals just to make sure that the ammonia levels or the nitrate levels don't get crazy. And they do. When you get 100 tilapia in there and they're reaching a pound, the water chemistry goes crazy. You've got to be on top of it daily. I do think that these types of projects, these PBLs, I mean, they're 6-month projects. So it really forces a kid to go through, long term and work on that. And they take it to heart. I mean, they don't want to see a fish die. They're invested halfway through this. They don't, these are their babies. They don't even want to eat them at the end of it. I have no such compunction. So
you're not so
A
attached.
vicious cycle.
right.
Yeah, they got to learn circle of life somehow. So
Yeah, and they're actually quite tasty. So there is that too. So
yeah.
I mean, here's something. Here's a thought of what you can do with the tilapia as sort of an experiment is to feed them different things. Have 2 fish tanks going, and we actually used a different source of protein to go along with the traditional diet, just replace part of it and to see if a non-fish protein had an impact. And that was one of our research projects.
that's pretty cool.
And
Yeah, I think
you get them as food.
I think you could do that with smaller fish in tanks.
They're hard to weigh individually.
Yeah,
Mhm.
yeah.
And you
well-
cannot identify them individually, so you have to weigh them as groups and chart the weight that way.
Right.
But we did them as fingerlings up to about 3 inches, and then we had to quit.
Yeah, well, with mine, I can't even catch them. They're way too fast. I mean, it's in murky water and it's 550 gallons, so trying to catch a single fish is impossible. You actually have to drop the water. You got to drop the water level way down. So, I mean, I guess if I had more room and I could do that, and if the agricultural biotechnology was more of my curriculum, I could probably do that. But we're just, it's all I can do to just pull off this one particular thing because it's so time intensive. I mean, I have to give a key, believe it or not, guys. I have to give a key to my students to come in on a weekend to feed and clean these tanks. Now that's problematic. So you have to have the right students. Of course, these are my seniors. These are my biotech 3 students typically that I'll entrust in a very small group. But yeah, I mean, it's constant. And when you're off on the holiday, you're still coming in and feeding and cleaning the fish or getting your kids to. And
Somebody's got to.
Somebody's got to. And the other thing is, I don't trust them more than 2 days. Even my students that really are good and about following the procedures and protocols, and they still just don't do as a job, the same job I would. You know what I mean? Because there's all these other aspects that we have to put. We have some chemicals in bags, filtration bags that we put over there just to take out some of the additional nitrates, and you have to wash those. You have to put them in Clorox water, and then you have to wash it all out. I mean, so there's, you start getting into these types of projects. If you're talking about a true agricultural, farm type situation, as y'all well know, in ag science, I mean, it's a lot. I mean, animals require a lot of attention, and you can't.
Yes, they do.
Yeah.
And so do greenhouses.
Yeah.
Right. Yep. You're tethered to it.
Yes.
You gotta commit.
Yep.
When we went to closing down the school and I was told you're not allowed to come on campus, I said, who's going to take care of my greenhouse? And I had my principal got named he was interim principal, got named principal the Thursday before this happened on Monday. And the district told him he was going to have to water the greenhouse. And I told him, no, he was not going to do it. I said, no, you've got all this other stuff. You're not in charge of the greenhouse. I am. Just let it be.
Right.
Yeah, just pretend you don't see me, and
Right.
fine.
That's right.
Yeah, wow. Right, so a lot of these turn into super long-term projects. So thanks, guys.
Mm-hmm.
And if you want to just share your emails in the chat, and y'all can keep up with each other's aquaponics and hydroponics.
Yeah, I'd love that.
Yeah, I love the collaboration.
I'd love to. Hey, any opportunity to go and see other programs and what you're doing, I think that's one of the big wins for the system here with Georgia Bioed, and thanks Megan and Kristen on that, is helping us to collaborate and maybe even putting together a day where we go around, and hit a couple of different schools and see what they're doing and just get those ideas. It's great seeing this and getting the recommendations on paper, but seeing it, nothing replaces actually seeing it firsthand. And the questions to ask at that point, I think sometimes.
Yes.
Yeah.
Agreed.
Thanks. Awesome. And I think before we went on the rabbit hole of aquaculture, Amanda was going to add something. I don't know if the thought has left her mind.
I was going to say hydrangeas. You can use hydrangeas or rhubarb to do a lab with pH, which goes into agriculture as well as chemistry and biology. When you talk about how plants grow in different pHs, soil requirements.
Yeah, that's a good point.
And you can find them just about anywhere. Yeah.
Mm-hmm.
Well, now isn't the pH affect the hydrangea and the bloom color?
Yep. Yeah, I think it follows the acid base thing. So acid red, base blue.
Yeah.
Probably would need to get some florist hydrangeas, the small ones.
Mm-hmm. Well, and I know you're not supposed to do it, but you can like cut off the blooming parts and then put them in water and like start rooting them basically. And then you could maybe adjust the pH of the water that they're in.
Yeah,
so
with acetic acid.
Mm-hmm. Yeah, so that way you could just get maybe one plant and then cut off the different blooms, because I think that would start to add up if you're buying multiple hydrangea plants.
Yeah, but I just think, how long would it take it to go to bloom, and then you could force it to bloom.
Yeah, you could force it with the hormones.
Or
maybe take the ones that are already blooming.
I've not done it, but I think it's like more of a it's going to be a long term.
Yeah, that's definitely not like a 2-day event.
Yeah.
Yeah. Cool. But that's a good point. So you can kind of tie together the agriculture with growing the hydrangeas, and then also that pH and chemistry, and then use it for general bio. So that's an awesome way to tie the three.
Also pH in food science.
Mm-hmm.
There is a lot of pH with food science.
Yeah.
As far as bottling and canning.
Yeah. Food science. Importance of pH. Yeah. Canning and storing everything.
Barbecue sauce. You can actually look up the food code through FDA and see what they require for the pHs. Maybe even be able to test the pH of different barbecue sauces and see what it is.
You said USDA?
USDA food code.
Cool. See recommended pH.
Oh, no, required.
Right, yeah.
If you're going to bottle something there's a required set.
Right. Cool. Those are all awesome ideas. I can't see it right now, but if you guys have anything else to add to the shared labs folder, feel free to share. I'll throw some more stuff in there that hopefully you guys can use. But thanks. Let's see. And then the last slide I threw in here, just kind of Kristen and I have been tossing around the idea with trainings of trying to get some other teachers in there to just see how we could get y'all to work together, you know, and hopefully we could facilitate that in the future. So we've been talking about getting English teachers in to start working on the writing. So Sarah Eels does, she's at the Paul Duke STEM Academy in Gwinnett, and they specifically have set up the STEM Academy where the literature teacher works with the science teachers, and they are always working on writing lab reports or writing something about science, communicating science instead of, you know, working on other kind of literature topics. So making it focused. One of my experiences, I worked really closely with the teacher next to me, and we were doing the block, we had a block gifted bio and chem class. And so I was teaching the bio section and she taught the chem. So we made sure for the respiration unit that it lined up with when we were doing the reaction types in chemistry, because I also taught chemistry. So we went over, basically I would make sure we talked about the equation for respiration in the biology class and that we balanced it in biology so that they knew like chemistry's not just in your chemistry class. And then she would use respiration to talk about combustion reactions and the general setup of those with bringing in oxygen and giving off water. So just kind of to like get you guys jogging your minds on that. It, you know, I know y'all don't have a ton of time, but if the time comes up, if you have a friend at your school that's already, you know, they're an English teacher or they're a different science teacher, maybe y'all can work together to try to make your lives a little bit easier, or at least pound things into the kids. So any other thoughts you guys have, want to share?
For our school, for the writing component, we kind of started it last spring, but it went away, obviously. But one thing that we're working on in the future is we're working with our English department to help we have post-AP classes, and part of them will be the biotech as well. But we're gonna have it be a required component to actually write up their labs and their research. And we'll publish like our own school journal basically. And then that way they have things ready if they do want to submit them to like the Journal of Emerging Investigators and things like that. So we do that. And then also we have an organic chemistry class at our school. So my genetics research kids, we team up with our organic chemistry teacher a lot. And he'll teach the kids how to use like the GC and mass spec and stuff for biology purposes instead of just chemistry. Because a lot of our kids, we either have had them both at some point or we probably will. So we kind of try and do some cross-curricular stuff with our other post-AP courses.
Awesome.
One together. You're supposed to be able to, we're trying to figure it out, but you're supposed to be able to identify bacteria based off of the esters in their cell membranes using the GC. So we're trying to get that together and see if we can we can use that as an alternative basically to sequencing. But it's a work in progress. We've got to figure out a lot before we get kids involved in it.
Yeah, but that would be awesome. So we'd love to hear if that actually materializes, how that goes.
Yeah, one day we will.
I know COVID is kind of getting in the way of that too.
Yes.
So, yeah, awesome. That's
I'd like to add the cross-curricular.
Yes.
I do like to integrate graphs and some basic statistics.
Yeah, that's a good point.
Doing the labs and try to get them thinking about ratios and things like that.
Right. Like I know that chi-square is written into AP Bio like already, but bringing in more statistics and analyzing, understanding statistics, that's awesome.
Well, especially with, I've got kids like in basic agri-science. Yeah, I teach 9th through 12th graders in that class. And just to show them where statistics can be used and where they might see it at. And sometimes they don't even realize that some of the stuff they see every day is a statistic.
Yeah, that's a good point. And with a lot of the COVID research, you can talk about that too, and how these studies, you can look at sample sizes and talk about, you know, is that statistically significant, or, you know, would you really need more samples to actually be able to say something? Because they'll see a graph and it's like 5 people responded, but like, okay, great,
Oh,
that's
yeah.
not really representative.
Or,
Yeah.
or the labels are all wrong on the graph. Well, you pull a graph from a different resource and go, if you'll notice that this data doesn't line up because it's from different things, and they put it all on one saying, anyways.
Yeah. Well, and then there was that graph where they did like the number of COVID cases, but somebody organized it by order of size and not by date. And so the dates were all like intermixed. It was horrible.
Well, somebody was posting on Facebook saying we should go to a year-round calendar, and that statistics showed that when they did it at 2 elementary schools back in 2005, that it actually increased the reading scores of the children. And I said, I want to see your data. And when I saw the data, one set was the first set of data, the beginning of 4th graders. Then the next year, it was all the 3rd, 4th, and 5th graders. And then then they did the following year when that first set would have already been in middle school. I said, you cannot compare this data and say it improved the scores when it is not related. But I mean, this was adults too that were doing it.
Right.
So just teaching them how to read that better is a good practice.
Yeah, and then highlighting the importance of peer review when generating data. Have somebody else look at it and evaluate your methods.
And also having them make the graphs with their data.
Yeah. And then showing, right, is it showing what you meant to be showing? Can you use that graph to stand alone and really represent what's going on?
And can you write about it?
Yeah.
We do the FFA Agri-Science Fair, and having the kids graph their data and then talk about it and extend all that. And it's hard even for the seniors.
Yeah, they don't have too much experience with putting thoughts into words and writing it, putting pen to paper with those thoughts. So that's awesome. All right. Thanks, guys. So I will, I'll definitely share out this whole PowerPoint with everybody. There are a couple other slides here. So next week we'll do some curriculum mapping by subject if you guys are interested. Or we can just do, if we want to stay in a big group, we can do some kind of contingency planning for maybe what can we make digital versus, what absolutely has to be done in person, or can you guys find compromises for those things? So whatever you guys want to talk about next week, and whatever we can do to make your lives easier. And then if you haven't added already, our new Instagram is down at the bottom. So the Georgia Bio Ed, we will start posting on that very soon. So feel free to follow us, and then stay tuned for our first view, or our first post. So cool. Lisa said contingency planning sounds good. So we will get together and rack our brains. And maybe we can pull together some resources. I've seen a couple things cross my feed as far as like recommendations go. So maybe putting together a list like that, just to try to pound some stuff into your administrators' heads too, as far as what is safe and what will be safe. So you guys have anything else you want to share today? Contingency planning. Yep. You're welcome, Lisa. Happy to help. So if you guys are good, have a great day. Feel free to email me if there's something you think of that you want to talk about next week, or if you have anything to add to this, definitely add to the folder, or email me if you want to add it to the PowerPoint. Thanks, y'all. Cool. Have a great rest of your day. And I'll see you next week.
Bye. Thanks, Megan.
Bye.
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 Bye. Have a good one.
You too.
Bye-bye.
Extra Amanda.
Did she call in too or something?
I don't know what happened. Sometimes Zoom is weird and does like duplicate things.
Okay.
So
I'll go
I
ahead
don't know.
and end it though.
I'm gonna save the chat. Okay, now you can end it.